Hello everyone, this is S. Divya from Biotechnica, and today I am here to give you some insights regarding biological membranes, their structure, function and membrane dynamics. So this particular topic is not only important for CSIR NET aspirants but also equally important for DBT exam as well as GATE exam. So let's dive into the topic. So biological membrane, what is its composition, the structure and function? We will look into one by one each. So when I say biological membrane, basically it is composed of three major biomolecules. Lipids in the form of phospholipids and sphingolipids. Proteins can be peripheral or integral membrane proteins. And carbohydrates exist in the form of glycoproteins or glycolipids. So each of this component has its unique importance to biological membrane. Looking into the structure of biological membrane, it's a lipid bilayer with hydrophilic head group and hydrophobic tail. So this is how it is. The polar hydrophilic head group which has the phosphate attached, and the non-polar hydrophobic tail, which is we call as a fatty acid tail. What is the major function of biological membrane? Well, they are selectively permeable. That means it will not randomly allow all the molecules to pass through it. Rather, it will allow only unique molecules, or depending upon the pore size, they will allow the movement. Cell signaling. So there will be a ligand whose receptor can be present on the membrane. So when this ligand goes and binds to the receptor, a signal transduction pathway will be initiated. So basically transmitting information from external world to the internal of the cell. Then of course transport processes. So molecules can move inside the membrane as well as go outside the membrane. So as we all are aware that the most accepted model for plasma membrane is fluid mosaic model, right? So if you look into this fluid mosaic model of the membrane, as I told you, it is a lipid bilayer and it will be having its head group towards outside and the tail portion, which is hydrophobic by nature, is towards inside of the membrane. Then certain positions you will find peripheral proteins are present. As the name indicates, they are located at some peripheral surface of the membrane. Like that, we have integral proteins located. It could be a single transmembrane helix or it could be multiple transmembrane helix. But they are strongly attached to the membrane, unlike your peripheral proteins. These peripheral proteins can also be connected to or covalently linked to some kind of lipids. For example, it could be connected to a palmitoyl group. It could be connected to a myristoyl group, or it could be connected to a geranyl, farnesyl, etc. Like that, we have an anchoring protein known as GPI anchored protein, glycosyl phosphatidylinositol protein. Then in between, you will also find sugar molecules present in the membrane, but they are not present as such. Rather, they are present in the form of glycoproteins or glycolipids. Then, in between the phospholipid tails, you will find sterol content is present. So, what is the significance of a sterol? Well, basically, cholesterol is present in the membrane specifically for maintaining fluidity and rigidity of the membrane. Okay. Next. So we will look into each of these biomolecules individually. So the simplest classification of membrane lipids could be this one. We can classify them as phospholipids and glycolipids. So how can we differentiate these two? Well, as the name indicates, phospholipids will have phosphate group attached to them, and glycolipids will have sugar molecules attached to them. These phospholipids can be further classified as glycerophospholipids and sphingolipids. Glycerophospholipid is considered to be the most abundant form of phospholipids. And the structure involves glycerol as a backbone, 2 fatty acid, and the phosphate group can be attached with any X group. Suppose it could be an alcohol or it could be any other molecule. We will look into that structure in the upcoming slides. Similarly, for sphingolipids, as the name indicates, we have sphingosine as their backbone. They will have 1 fatty acid chain attached. Phosphate also will be there, which will be connected to any X group. For example, here it is mentioned choline. So, depending upon the X group, the name of the lipids will vary. So, that is about our phospholipid classification. Now, if we look into the glycolipids, again we have sphingolipid. Now, a question can arise. How can you differentiate between a sphingolipid that comes under glycolipid and a sphingolipid that comes under phospholipids? Well, we can call it as a glycosphingolipid, phosphosphingolipid, or sphingophospholipid, sphingoglycolipid. So, whenever any question comes related to that, it will be clear whether they are asking glycolipids or phospholipids. So, like your sphingophospholipid, here also we have sphingosine as their backbone. But what is the difference over here? You will not find phosphate group attached to any X molecule. Rather, you will have a sugar molecule attached. It could be either in the form of a monosaccharide or an oligosaccharide. Another classification is galactolipids, which is otherwise known as sulfolipids. Now, why it is known as a sulfolipids? Because of the attachment of a sulfate group to the sugar moiety. And like glycerophospholipids, here also we have a glycerol backbone present. So, more or less we can say that quite structurally similar to lipid molecules are glycerophospholipids and galactolipids, and these two are similar. The only difference is the presence of a lipid component and the sugar molecule. Next, this is the glycerophospholipid structure. As you can see, the head group is present. It is having a fatty acid tail, and this is the glycerol backbone will be there. So, if this X group is H, we call it as phosphatidic acid. If it is an ethanol amine, we call it as an phosphatidyl ethanol amine. If it is a choline, which we have seen in the image, it will be phosphatidyl choline. If it is a glycerol, phosphatidyl glycerol. And if glycerol is itself an X group, we call it as a phosphatidyl glycerol. Then, if it is an inositol as an X group, you have got phosphatidyl inositol 4,5-bisphosphate. If we take this entire phosphatidyl glycerol as an X group, what we can get? Cardiolipin. And cardiolipin is one of the important glycerophospholipid present in the inner mitochondrial membrane of cardiac heart muscles. Next. So, this phosphatidyl choline and phosphatidyl serine is otherwise known as lecithin and cephalin. Okay. So, choline and serine as the X group, we are getting phosphatidyl choline and phosphatidyl serine. These are otherwise known as lecithin or cephalin. Next. Similarly, sphingolipid structure. So, as you can see, we have a sphingosine as the structure and there will be an X group present. So, if it is H, we have ceramide. If it is a phosphatidyl choline, it is a sphingomyelin. Then, depending upon the glucose molecule, different sphingolipids are present. Okay. Now, what are the most predominant phospholipids that are present in the inner and outer leaflet of the plasma membrane. In other words, which is present towards the cytosolic region and which is present towards the extracellular side, that is outside of the cell. Remember, phosphatidylserine, phosphatidyinositol and phosphatidylethanolamine. These are the three major lipids present towards the inner leaflet of plasma membrane. Similarly, sphingomyelin, phosphatidylcholine will be present towards the outer leaflet of the plasma membrane. So maybe due to some emergency situation like a wound or apoptosis, you will find that this phosphatidylserine will be switching to the outer leaflet of plasma membrane. Now that is being catalyzed by a separate group of enzymes regarding which we will see. Okay, so be clear with the phospholipids that are present in outer and inner leaflet. Next, what is membrane dynamics? Well, membrane is dynamic by nature. That means what? It is always moving. The phospholipids that are present within the membrane, that is plasma membrane, can move to different parts of the membrane with or without a catalyst. So what are the factors that can affect these kind of membrane dynamics? Remember, if there is a shorter chain length or shorter fatty acid chain with higher number of unsaturated fatty acid, in that case you will find that membrane permeability as well as membrane fluidity will be increased. But if there is a cholesterol accumulation present, remember the permeability will be affected, it will be reduced. So remember one fact that the presence and absence of cholesterol will definitely affect the membrane dynamics. As you can see, cholesterol make a increased fluidity at low temperature and increase rigidity at higher temperature. So we can plot a graph based upon this fluidity and temperature. As you can see, as the temperature increases, there will be greater fluidity. Right. So if you look into this picture, why it is having greater fluidity? Because of the degree of unsaturation. That means unsaturated fatty acids are those which are responsible for the formation of kinks. That is a kind of bend because of which the space is available and because of which the fluidity increases. Whereas on the other hand, we can see that because of the presence of saturated fatty acid, the phospholipids are closely packed, because of which the membrane fluidity decreases. Next, how exactly this cholesterol is present within the phospholipid bilayer? You can see that it disturbs the close packing of the phospholipids. That's the reason they are responsible for regulating membrane fluidity. This is how the cholesterol is inserted within the phospholipid bilayer. Now, what kind of interactions does takes place between them? So, this is the structure of the cholesterol molecule. So, this is the OH group present and this is the hydrophobic region. So, the hydrophobic region of the cholesterol molecule will be adjacent to the fatty acid chains of phospholipids. Similarly, the OH group of the phospholipid molecule will be forming hydrogen bond with the phospholipid head group. This is how the cholesterol is inserted within the plasma membrane. Now, as I was saying you, the presence and absence of cholesterol will influence the membrane dynamics. That is membrane fluidity. Why? Because cholesterol function as a buffer, preventing lower temperature from inhibiting the fluidity and higher temperature from increasing the fluidity too much. So remember this concept, at lower temperature It prevents close packing of phospholipids, because of which the membrane fluidity increases. So we can say that here the degree of unsaturated fatty acids are more. Similarly, at high temperature, there will be constraint in the movement of phospholipids, and we can see the membrane fluidity decreases. So in this case, what we can say? Saturated fatty acids are more. So this is how the cholesterol with temperature is regulating the membrane fluidity. Now, as I was mentioning, the movement of phospholipids takes place within the membrane. So it can be different types. For example, it can undergo flip-flop diffusion, which is not catalyzed by any enzyme. For example, this is a phospholipid here. It can flip towards other leaflet, but it's a very slow process. But if you see on the other hand, lateral diffusion means adjacent. That means on the same lane. As you can see, this is your phospholipid diffused towards this direction. So compared to the uncatalyzed transbilayer diffusion, uncatalyzed lateral diffusion will be faster. Now, which one is the fastest? If it is being catalyzed by certain enzymes. For example, flipases. These are the flopases and scramblases. These are the three major transporter enzymes that are responsible for translocating the phospholipids from one leaflet to other. As I told you, you have got specific phospholipids present in the inner leaflet of the plasma membrane and specific phospholipids present in the outer leaflet of the plasma membrane. So, it is this flipases with the help of P-type ATPase pump will be flipping or taking the phospholipids towards the inner leaflet. Remember, flipases, I stands for inner leaflet. So, phosphatidyl ethanolamine and serine can move from outer to cytosolic leaflet. Flopases, which is carried out by ABC transporters. Flopases, O stands for outer leaflet. So, the phospholipids will move from cytosol to the outer leaflet. Remember, these two processes are ATP dependent, whereas scramblases are ATP independent. So, as I told you, phosphatidylserine normally resides in the inner leaflet. But in some emergency situation, it can also flip to the outer leaflet. Now, that process is carried out by the enzyme scramblases, which requires calcium ion binding for their activity. Next, the proteins. So, we have two types of proteins. So, that is all about the lipids. Next, we have proteins. Peripheral membrane proteins can be attached to either surface of the bilayer. How? By means of non-covalent interactions. It could be electrostatic or hydrogen bonding. That's the reason it can be extracted by any kind of mild treatment. Whereas integral membrane protein are firmly associated with the lipid bilayer and held together by hydrophobic interactions. That's the reason they has to be removed with those agents that can interfere with the hydrophobic interactions. For example, detergents, organic solvents, denaturants, etc. So, we have different types of integral membrane proteins based upon the orientation of the protein. Like you can see type 1 and type 2. These two are opposite to each other. In what sense? See, in type 1, N-terminus is facing the outer leaflet. Whereas in type 2, N-terminus is present towards inside. Type 3, you will find multiple transmembrane helices are there present within a single polypeptide chain. Whereas in type 4, you have individual transmembrane domains and they will come together forming a channel. Type 5, they will have a lipid anchor. Type 6, they have a lipid anchor, along with that a transmembrane helix is also present. So number of examples are present for each type of this membrane proteins, that means integral membrane proteins. Next is membrane carbohydrates, which contributes very less, that is around 5 to 8%. Predominantly glycoproteins exist in the form of cerebrosides and gangliosides. Glycosylation takes place at the extracellular surface of the membrane, and these carbohydrates are mainly responsible for cell-to-cell recognition. The major role of this carbohydrates is of the glycolipids present within the RBC, plasma membrane. So that will determine whether a person is carrying type A, B, AB or O as a blood group. Remember, O antigen is the parental from where A and B is obtained. So it is a combination of galactose fused with fucose, N-acetyl glucosamine, galactose and glucose. So if a person is having A antigen, the same parental molecule but one extra N-acetyl galactosamine will be there, catalyzed by N-acetyl galactosamine transferase. If a person is having B antigen, then the same composition like O antigen, but instead of N-acetyl galactosamine, here galactose molecule will be added. The enzyme responsible for this is galactose transferase enzyme. Next, the last part of the session is all about lipid rafts. So these are specialized microdomains present within the cell membrane, enriched in cholesterol, sphingolipids and specific proteins. These are the organizing center for the assembly of signaling molecules, influencing membrane fluidity, protein trafficking as well as receptor trafficking. So as you can see, the visualization, the difference between a raft region and a non-raft region. So raft region, you will find that it is enriched with saturated fatty acid. So the fluidity will be less, proximity will be more compared to the non-raft region. And you will also find abundantly sphingolipids are also present. So if you look into the composition, 3 to 5 fold the amount of cholesterol found in the surrounding bilayer. As I have shown you, enriched in sphingolipids in the form of sphingomyelin, glycophospholipids which are typically elevated by 50%. So what is the role of cholesterol over here? A glue that can hold the rafts together with the plasma membrane. Phospholipids will be present in the form of saturated fatty acids. GPI anchor proteins can also be seen on lipid rafts. What are the properties of lipid rafts? These are detergent resistant, that means remain intact even if the membrane dissolves. That's why we say that if cholesterol is present, rigidity and fluidity is maintained or regulated. It can move across the membrane, can join together or break down into smaller rafts. Types of lipid rafts, we have two types. One is a planar lipid raft which is non-caveolar by nature. Another one is a caveolar. Planar rafts are continuous with the plane of membrane. So you cannot find any distinguishing morphological feature. Whereas caveolar on the other hand is a flask-shaped invaginations like this, and it contains caveolin proteins, most readily observed structures in lipid rafts. So the way caveolar is having caveolin protein, planar rafts will be having flotillin proteins. So both of them are expressed abundantly in neurons, nervous system. So caveolins are found in brain, microvessels of nervous system, endothelial cells, astrocytes, oligodendrocytes, Schwann cells and many other areas. Whereas these flotillin proteins are found in neurons wherever caveolae are absent. So that's it. So I hope so I was able to give you some good insights regarding the biological membrane. And for more details, you can always enroll into Biotechnica, where we will have more elaborate lectures regarding all the topics of NET syllabus. Thank you.